looks similar, and the sequences are pretty thick.
It's behind that one, I believe.
And we are
that's quite a width.
60 or so kilometers east of the active volcanic arc. This does not look like airfall to
there
me.
It is.
Unless there was a huge explosion back in previous history of the arc when volcanic centers might have been located out in this part of the region between the trench and the currently active arc.
Okay, Tara. Harsh.
Here's my new bamboo coral for the day.
Wow, really?
Lyrate. Yeah,
Okay.
so this is one that Les and I, if we'll get a little closer, I'm pretty certain though. It's got a very characteristic structure. We're looking at it kind of on the side, but if you were to look at it from another plane, it would look like this candelabra or this lyrate form. And Les and I are describing this as a new genus. We've collected it in several places and seems to always have this characteristic growth pattern that it grows like a whip for a while, and then it trifurcates. So at one stage it looks like a little trident, and then it continues to put out branches kind of in pairs on the far distal sides of the colony to like the right and the left to create this kind of candelabra-like form.
All right, to zoom more.
Yes.
Does this look like any of the species that you've been working with? Kind of hard to tell.
Yeah, well, so far there's only one species that we know of, and so yeah, we couldn't tell, I don't think, without
I have a bit more
doing
zoom.
genetics on it.
I don't have much more stability.
No worries.
Gotten myself into
This one looks a little bit more raggedy
Understood.
than some that I've seen. But we've seen this morphology up in the North Atlantic, in the Bahamas, around the Hawaiian Islands, so it seems to be pretty common and able to get around. Assuming that it's the same species, which, you know, I can't say that for 100% certain, as you've just pointed out.
Like right on its center stop.
And so the only way you could tell this would be you doing
Let's see
genetic
how it goes.
tell if this is a new species would
There's
be
nothing
if
wrong
you
with
did some
moving around
genetic work
coral
on it.
either.
That is correct.
Just
really.
informative.
Wow.
Great.
Yeah, this is perfect.
This is some beautiful imagery.
Certainly is. I don't see any sclerites sticking up between the tentacles.
Okay, let's pull out.
Don't
Okay.
think. Oh no.
Do we have a couple more seconds?
Sure.
It's just such a nice shot.
Power and ventilation secured at this time. Power and ventilation.
All right.
So Les Watling is commenting that this is very nice, pretty nearly full grown, but
Thank
strong
you.
and growing tips on each branch.
For that?
Sure.
Lasers are on.
Just put it right at the base of the coral. That way it's the same. And that can be used for measurement later on then. Maybe I can sweep it up across the...
Okay, pilots, I would say ship has completed the move.
Roger that.
There we go.
Nice.
Okay, lasers
Okay.
up the hill.
Up the hill. Rock on.
It's a tough day.
Oh, look. This
Oh
thing is
my
so calm
gosh,
here.
look at this.
Oh,
Wait till you
beautiful.
see this.
Scott, you're going to be in
Scott
heaven.
and Les. Oh my
Heaven,
gosh.
heaven, heaven.
A Metallogorgia garden.
Yep, indeed.
I was just about to go back to making my salad, and then you made me
Forget
come
it,
back in the
Scott. You're doomed.
Yeah, that's great. You can see there's one of the more juvenile stages at the lower part, and then you have 1, 2, 3, 4, 5 adults, it seems like, on the upper part. And you notice they're all about the same height. We see that consistently all over the world. And each of them has only one brittle star ophiuroid in its branches. And you also see that there's nothing else living on these colonies. There's no barnacles, there's no other kinds of brittle stars, there are no crabs, no shrimp. And so the hypothesis is that that single brittle star that's living there with its very long arms wrapped all around the branches may be getting rid of larvae and other animals that settle on the colony. And perhaps there's also some biochemicals that are being produced by the colony that are noxious to other organisms that are landing on the colonies. It's hard to say, but this is a very consistent observation we make. Once in a while, we'll find a colony that has the tissue removed from part of the skeleton.
Okay, well, you want to
And if
angle
that's
back.
the case, then you might see barnacles, crinoids, and crabs and things, snails on the skeleton, but only where there's no
What
tissue.
depth is the end point
So
at?
there must be some kind of secondary metabolite or something going on.
That might be the result of,
Raised this interesting point just a few minutes ago that perhaps the interaction of the brittle star and the coral causes one or both of them to be releasing some kind of chemical that discourages other animals from settling on the colony. Anyway, that was beautiful, fantastic to see, and it's really interesting that not uncommonly when you find one of the Tapagorgia,
It's a little more than 100 meters left to the... We could always head
what do
towards
we
the
know about
waypoint
the
once we get to
reproduction
the top.
of this
Or get to
genus,
that
or in
depth.
fact about the whole
Yes,
family?
if you consider we've made 80 meters in 50
Yeah,
minutes.
we don't know very much, but let's have a PhD student, Ann Simpson, who was working on some understanding some of the reproductive strategies. And one thing that she did determine is that each colony is either male or female. So that's kind of interesting. So that means that the colonies presumably have to be living close enough together that the male sperm can swim to the eggs of the female. Beyond that, I don't recall that she learned anything about the larvae, whether the larvae were capable of swimming or if they just crawled or something else. Perhaps Les could put something in the chat room about that.
And I am having a hard time keeping up with the changes in the rocks outcropping on this wall. We just came over at 1751, contact with a more coarse rubble, and then we're now at 1748, and back to this more massive, finer layered material, and above it,
These big blocks again.
Everything is fractured up. I don't understand why the stuff below isn't fractured. I am confused, but it's very cool. And I'm never going to get lunch here.
Oh, forget it.
This is too interesting.
You missed lunch.
Oh, gosh. Oh, look at this, Shirley. The layering. Don't look at don't look at the coral. Look at the layering. Gorgeous layering. Oh wow.
Might be an
This
unusual
is
sea star
amazing.
in the back.
Help. Can't write fast enough. I know how to slow it down.
Tara, let's take a look at this layer.
And he
Sure.
goes, let's take a look at this layer. Why she wants to do this, I don't
We can
know.
look at the starfish. We can look at the starfish.
No, well, let's look at the layer, just the whole layer first.
Okay, there you go.
then- Yeah, actually, if we pan to the starfish, we can get a good close look at the layer at the top.
It's not quite full zoom.
Thank you, pilot.
You're very welcome.
Boy, you know, it's really hard to tell whether this is a rubbly layer or not because it's got so much manganese oxide on it that it looks like this cauliflower surface. But the overall appearance looks as if there are a series of layers that are buried
Sure
underneath
does,
the
yeah,
manganese.
yeah.
So this was exposed a long time ago and has got a real thick coating of that. Okay, pilot, you can, if you wish, zoom back to the star on the left, or look at the yellow thing on the right. Your choice,
Have
or
we seen
Shirley's
it?
choice,
Have
or
we-
the shortest
okay.
choice.
Where would you like to go? Shirley?
Shirley, you want
we're
to
good.
take a
Take a
you
quick,
want to take a
quick,
look at the star?
quick zoom at the star.
Star.
Okay, a choice is made.
Star tis.
All right. Do we have our lasers on?
We do not.
Could we have our lasers on?
Got them for you. Lasers on.
There we go.
So the lasers, the two red dots are 10 centimeters apart, and that helps us get a little bit of an idea of how thick the layers are, and they are probably about a meter and a half from the bottom to the top of these two major layers.
Nice fat sea star.
Fat, so
Well,
it's full
not
of
too-
food.
I'd say semi-full.
Mm-hmm.
Right, I have a bit more zoom.
We don't have to.
Oh boy,
Yeah,
really
you
thick
need to start
manganese
moving
around
soon.
it too.
No, and, need to get moving.
That's good video as soon as you're happy with it.
Roger.
For wide.
Okay.
And we'll move up.
And
Okay, pilots,
overhang.
Looks like we're in position.
Roger that, Nav.
Wow, alternating thin and thick layers of rock. Very horizontal. Our heading is generally due north. Hard to tell exactly what
Wow.
these layers are made of because they're so
These
heavily coated with manganese.
Steps.
Looking
Look at
straight
these
down.
steps.
Beautiful stair steps again.
Wow.
More or less right ankle fractures exposing these. This is so
Looks like I'm
completely
gonna make a
different from
jump right
yesterday.
Yeah. Nice little flat spot.
Suddenly there's some current. Poke my head up above the ridge here and whoosh.
Looks like it's coming down slope.
Yeah. Certainly. from
Oh,
left
yeah, a
to
little
right.
bit. Yeah, I can see it now.
And our current is back, folks, moving from left to right. You can see the stalks bending over toward the right-hand side.
Wow, look at that. Look.
So, Patty, I've had a bunch of calls on people in here, and I've been distracted. But did you mention what the heck we're looking at?
I
I'm not
sure
getting what
tried.
these layers are.
I sure tried, Chris. As we zoom in, everything is just totally covered with manganese incrustation that looks like it's pretty old, and we're looking at botryoidal surfaces. Initially, I was wondering, because the layers showed graded bedding, that this might be volcanic clastics, but we're so far east of the volcanic arc, it's hard to imagine that we are looking at an accumulation of a tremendous thickness now. I think we are now, I've been trying to keep track of the height since we crossed over a very distinct change
now
in
look at
morphology.
Yeah, we are really oscillating.
But we're at least 50 meters of this material.
Pretty good spot.
All right, now
Yeah.
look at the one over on the left there. That looks
The
different.
the places where a lot of the surface of this wall has been knocked clean, I would say, of manganese oxide coating, what we see is a very pale brown material that looks, when it is looks like it's fractured in place, broken up. It looks like coarsely granular sediments, basically. But the layering in this part of this scarp is spectacular.
Well, this is a pretty naive question, but
No.
how is the seafloor here old enough to accumulate this much manganese crust on it? Because we're on the west side of the trench, right?
Yeah, right.
That's true.
Okay.
Yeah, but we don't
Yeah,
know
yeah.
when
Probably
the
not a-
fracturing
probably
that
not a
produced
good
these
idea.
fault walls that we're diving on occurred. And according to results of DSDP drilling south of here at 18 North, there was in the late Eocene, early Oligocene time, which was from like 47 to, well, more like 35 to about 25 million years ago, a tremendous amount of vertical tectonism, so faulting, down-dropping, uplifting in the Mariana Forearc. I'm wondering if this could be exposures of that sort of thing, and that overlying material may have slumped off toward the trench to accumulate there, because in the later periods, from the late Oligocene through basically the Quaternary, there are sequences of maybe 400 meters thick of turbidite flows that have just been shuttled down toward the trench at 18 degrees north in depressions. We are sitting at the edge of a depression. So maybe a lot of material got sloughed off above these sequences and moved toward the trench. So we'll see when we get up to the top of the ridge what things look like. That's about as much as I can tell you about what we know about this part of the forearc, Chris.
We biologists are liking this depth.
You do, I can tell.
We're liking this depth.
This
range.
depth and this.
Fascinating seafloor.
Now, I hope you're going to zoom on this animal that looks like something out of Avatar, with a series of fan-shaped, almost coiled arms that.
Yeah, but I'm thinking.
Oh man.
Okay, pilots, just to
I
confirm
think we
again,
saw
in the first
something
hour
like
we
this on
covered
Leg
110
1.
meters.
This is beautiful.
We have 2.5 hours
This is
left
our
to do
first
270
view of
meters, so we're
the
more or less okay.
Chrysogorgiidae
Roger.
genus Iridogorgia, one of the most beautiful
Take a
octocorals
bit more
that you
zoom.
can see. And so this is in the same family as the Metallogorgia that you see on the left and the Chrysogorgia that we collected earlier. So it has that same characteristic that the polyps has, how do you describe them, look like a cherry blossom tree before. They look like these little pink
It's
droplets.
not quite full. I'm going to hold
So
there.
it's very thin tissue, but you can see that this skeleton has this main axis, has a spiral to it, and then there are these unbranched side branches that come off of the spiral, and the polyps sit on there. And very often we see shrimp associated with these colonies, and very often it's a female shrimp that's carrying eggs, and we'll see if that's the case here.
Yeah, there's a shrimp on there for sure.
Yeah, so I know in the North
yep.
Atlantic we see these shrimp, and they have purple-colored eggs underneath the abdomen, and I'm wondering if that's what I'm seeing here.
It is dark under the abdomen, darker than at the.
More purple than red.
Yeah, for sure, more purple than red.
Yeah, so those are probably the eggs. They hold them with the pleopods, or the swimmerettes, which are the little swimming legs under the abdomen. We have an interesting story from the Caribbean where we see one of these shrimp on these Iridogorgia, and we took a sample of the Iridogorgia, so we clipped the top of the colony, and when we did that, the shrimp swam away
Coming
from the
out for
colony,
us and establishing.
and the ROV guys wanted to try to chase it to catch it. Of course, they couldn't. It's very hard to catch a shrimp swimming through the water column. It's like chasing a butterfly. So we let it go. We then, we were at a relatively shallow depth, brought the ROV up on the deck, unloaded our samples, and then went back down to the same place, and we saw the shrimp back on the same colony.
Oh my!
Suggests they are pretty interested in being on a specific species of coral.
Okay.
Oh, that's fantastic.
Thank you.
Well, these little steps
Okay.
certainly are making
Looks
home for a
like
garden.
right now our
corals.
upslope course is 020.
Look at the next one too. You can see
this
there are
may
tons
just be
of
a
them sitting
buttress
right,
that
and they
comes
just
out
love
and,
right at the edge. They like living
but-
at the edge.
That tends to agree with the contours.
Roger that. Okay. And maybe we should get another move in then.
So
try
what are
that
all the rest of
020.
these stocks we're looking
Obviously,
at
this is
here,
away
Shirley?
from
Are
the.
these?
Yeah, from the. Well, you want to check with the
Oh,
watch
just
leads
dead.
and see if that
Yep.
end
Okay.
point is really important or going up slope is.
Yeah, because we could push.
Yeah, we can.
We
yeah.
can push that way
Uh-huh.
if they like. So yeah, let's check with watch lead. That's a good idea, Don.
Who's going to do that?
Nav is.
Okay, just checking.
Watch lead, this is Nav.
That'll be you, ma'am.
Watch lead, we're getting to a situation now where to follow the path directly up, we're going away from the end point. Which is the priority, to go up the slope or to
There's
head
sort of
towards the
a
end point?
leggy coral
Either
in
one's
the
fine.
back right center right now. That might be something we haven't seen before. I wonder if we could take a peek at that if it's possible.
Okay, copy that. Thanks, watch leads.
Wow. A lot of life down here. In these stair-stepping
Did you
blocks
copy that, pilots?
of beautifully
I
layered
did.
rocks.
So we should get the move
That's
underway then,
poetry.
020. Let's see how
Does
that
it
works
rhyme?
out.
20 meters again?
I think so, yeah. That seems like a nice, comfortable thing, you know, as we, sometimes we're getting in pretty close to the wall.
And we
With
seem to
Sirius,
have the right
right.
pace anyway.
Yeah.
Bridge, this is Nav. Could I have another move, please? And this time we'll have a range of 20 meters, a bearing of 020, and a speed of 0.2 knots.
If we
Good
have a
copy,
minute
Bridge.
here while we're getting oriented, looking at all these beautiful corals in the landscape, I just like to say something about the scientific method. We've been getting lots of good questions about reproduction, for example, in these corals. How do they reproduce? Are these males and females? And
Different
I think
crinoid
it
down there.
leads into an interesting discussion of how it is that we learn some of the various facts
Thank
and
you, Bridge.
answers that we're getting, or giving, excuse me. And one thing to consider is it's maybe fairly easy to come up with a question. It then becomes difficult to figure out how to answer that question, especially when you're working in the deep sea. And so when you want to know, well, how do those
It's
things
like you may
reproduce?
be
How
close
often
to
do
the
they
end of your
release
leash
sperm?
there,
Do the
Jeff.
sperms swim
Start
away?
moving quick enough.
Are the eggs shed
Sorry.
into the water column,
Then we
or are they
could,
held by the coral, and the
probably
sperm has to
extend
swim
that
to the
move an
coral?
extra 20
So on and
meters.
so on. All of
So
these
near somewhere,
require
looks like we've got
different
into
methods
the
of sampling and timing
flat
of
spot.
sampling and so
Okay.
on in order to be able to
Nav, you
to
copy?
answer
We
it.
want
And
to
a lot of
extend
it
that
boils
move
down
another
to,
20
we
meters.
just need a lot more time in the deep sea and effective sampling
Bridge, this
tools.
is
And
Nav.
Patty, I'll bet you would say the same thing for the geologists. You know, we need to
Hey,
have
Bridge, could I add
more
another
rock
20 meters
samples
to that
and
move,
sort of
please?
in situ visualization to see exactly where the rock is coming from and how it's oriented and so on.
Good copy, Bridge,
So,
thank you.
You know, these Okeanos Explorer missions are fantastic because they give us these observations and they just kind of tantalize us with more questions, and then we go back to the drawing board and figure out, okay, how are we going to answer those questions?
And the nice thing is that with some of the samples we've collected, the fact that they've been preserved, and so, you know, you might be able to go back in and
Looks like
do
a
some
tiny little
work, some histology, and see
one of
some of
those
the
corals.
reproductive tissue. And then knowing when we collected the sample might give you some insights about, you know, whether there's any kind of seasonality, whether they're hermaphroditic, whether they have separate sexes,
Copy
and
that,
so on.
Rich.
So it, you know, all these little, even though we're only collecting a few samples, the samples we collect are really valuable in terms of being able to at least try and formulate our hypotheses, the questions that we want to address even better.
And the other thing too is even the geology operates the same way, that we've seen sequences of different kinds of rocks exposed in the surface, and this will help returns with a submersible, for instance, and know exactly what depths to target to go and take
I'm not sure.
discrete samples from to categorize what we've been looking at. And what we're looking at right now
Counting why?
is a little white, what was that?
think it was an anemone.
That was an anemone? Shirley loves to pronounce that
Snapping
word.
again.
Sorry,
Oh,
what was
yeah,
that
I was
part?
muted. Anemone.
Oh, sorry. She was going, I'm an anemone.
You want to snap in again?
Oh, sure. Sorry, I didn't hear you, Jeff.
Sorry, I'm probably speaking a little too quietly.
There's
There
our little
we
anemone.
go.
And it is about 3 centimeters in diameter. Your 2 red dots are our lasers, and they're 10 centimeters apart, about 4 inches. So this guy's about 1.5 inches.
Okay, full white.
We are.
They look so much bigger on the
I know
screen.
they do.
We really
Yeah.
do. We get such a good
Oh, my
zoom on
gosh.
these things.
And when we get them up to the
so-
surface, they're just the teeny tiniest little things.
Can I get there, Dom?
Yeah, I think so.
All right.
We are broadcasting live from the NOAA ship Okeanos Explorer, America's ship for ocean exploration, and we are coming to you from the Marianas Trench. We are on a feature that we have named Mariana. Oh, sorry, she keeps correcting me on this too. Mariana Trench, not the
Yeah.
Marianas Trench.
Like not the Aleutians trench,
Not the
or
Puerto
the Puerto
Ricos
Ricans
trench.
trench.
Mariana Trench. Thank you, thank you. And we are on a feature called Explorer Ridge Shallow, because we did Explorer Ridge Deep. I'm just going to say that this is way more interesting today for us biologists.
And
Although probably Bruce would disagree with me, because yesterday
for
they saw
us
the most
geologists.
primo, I saw the most primo fishes yesterday, and apparently there, I think, may have been a shrimp that had never been seen alive.
yeah,
Also, so we still see
with
some
leeches
very-
on it.
I'm Shirley Pomponi.
Zoom partial.
I'm from Harbor Branch Oceanographic
Sure.
Institute at Florida Atlantic University, and I am the biology science lead, and I'm joined by...
And she is
different,
joined by
sea
her
star in the
geology
back, too.
science lead, which is me, Patty Fryer. I'm a marine research professor at the University of Hawaii, Manoa, part of the Hawaii Institute of Geophysics and Planetology on the island of Oahu. Come visit us. It's warm.
So, Scott and Les, was this one of the ones that you said earlier that if we saw another one, we should collect it? I don't hear anybody, so, okay, so probably not.
Hi, Shirley. Sorry, I was in the other room. That was a branched one that we saw, so I don't think this is the
Okay,
same, or
no.
we wouldn't know because this one isn't branching.
Okay, sorry. Thanks, Scott.
Okay, pilots, we are 30 meters into the 40-meter move.
Copy.
This
Roger that, and we are
expedition
moving in on you now.
Okay.
was planned and is being executed by NOAA's Office of Ocean Exploration and Research.
Don, you reckon we should just keep that moving?
And the ship is operated by NOAA's Office of Marine
Yeah,
and
it doesn't
Aviation
look like we've
Operations.
got a hard return
And
there,
the
so maybe the slope is
ROVs,
changing
the Deep
now.
Discoverer and Sirius,
Yeah, tack
are
on
being
another 20.
operated, maintained,
Let's
and
see what happens.
upgraded
I don't want to go 40.
by
Know what's up
the
there.
Global
That sounds good.
Foundation for Ocean
Based on the contours, it looks
Exploration.
like
I don't know,
maybe
did this
heading more
crew
north,
introduce
but,
themselves?
They
or-
did when they
did?
came
Okay.
in from lunch.
Okay, so I was at
Zoom
lunch
partial.
then.
Yep.
Yeah,
Here.
let's stay at the 20 degrees and
Yeah,
add
moving
another
on.
20 onto the current move.
Oh,
As
isn't
we
that
arrive
pretty
on-
beautiful.
gorgeous.
Isn't that pretty?
copy
Well,
that.
or just tell him to tack it on right now. I think we're looking good.
Okay,
He has not stopped yet, correct?
correct. He has not
Okay.
stopped.
Wow, this guy's different.
Yeah,
It is.
looks like he's.
It's so much
Okay,
fun to
take a
see
bit more.
something
Could I
that
add on
you know
another
the
20 meters to
biologists
the
are
last
saying,
move,
"Wow,
please? So that's another
this
20
is a new
meters at a
I'm
bearing
trying
of
to take the lasers
0.20.
up over it.
tiny starfish, sea star.
Sea star.
It's not a fish."
Good copy, Bridge. Thank you.
Looks
Yes, we
good.
are. Bailout 180.
Is it alright to zoom a bit more, pilot?
It's got
Yes.
a relatively long cirri.
Holding.
It's kind of attaching. So this is one of the unstalked crinoids. We've seen a lot of them that are on long stalks. This is one that's unstalked, a
Actually, Shirley, I
commatulate.
think it is a stalk crinoid.
You
The
think so?
stalk's
Really?
behind him. We just can't see it.
Really?
It was fooling me for a while. I think this is something called Proisocrinus.
Really? Boy, it has a tiny
Oh, that
stalk,
is
huh?
cool.
Go for a while. I'll try and come in close again. I drifted away.
Oh boy, what we're seeing right here in the middle of the screen is a change in the angle of the bedding. It looks like either a block has broken free and slid down slightly to the left, or we're looking at a fault. Hard to tell. In the Sirios view, you can see what looks like sort of a curving of the rock layers in front of the ROV. Sort of curving
I'll
away.
be
So
darned.
if you
Look
can
at that.
the way
Yep.
it's attached to the
Yep.
rock there, that kind of little finger
Yep, you can
type
see.
of attachment where the
Really?
stalk.
Oh
Yep,
yeah, you
yep.
can see.
You can
Yep.
see it now.
Yep, there it is. Oh, that's a perfect view.
Yep, you can see that. I thought before when I
Showing
saw
it
that,
off
Chris,
there.
that it was another little, like a little sea star under there, but that's the base of attachment. Wow, that's totally cool.
Chris, can you tell us again?
Okay,
Did you
Jeff,
say the
when you
species?
start moving.
I think he said Proisocrinus.
Yeah, so this is almost certainly in the genus Proisocrinus. The sort of angular stalk on it, the way it's holdfast there, the bottom of the stalk, the very red arms on there, and the number of them, and then right underneath the calyx, there's going to be some small cirri, these little projections that come out right before the arms come out. All of those point directly to the genus Proisocrinus.
Oh, Shirley, look at all of these beautiful critters just hanging on for dear life on the
I
edge
know.
of these layers. Oh, and look at the top.
Okay, Chris, go ahead. You can make your pun now.
There's a forest.
He's going to make his pun. It's a biology pun. Go ahead, do
Okay.
it.
Wait for it.
You mean the one I put in the chat room already? No, I can't possibly say that again.
Aha.
He called it a heavy metallo band.
Usually when we see such distinct undercutting of the layers, it means that whatever that layer was softer than the layers above and below it and more easily erodable. So just different grain size, different composition. And these wonderful things seem to like the softer substrates. I'm surprised.
Wow.
Well, there's something funny that's sort of green right above the lasers right now. I wonder if we could take a look at that.
Green thing
Yeah,
above the lasers.
actually, you see it.
huh?
It's right below them now.
Down
It's
below,
right, yeah, it's
yeah.
right below them now.
Almost on it. Yeah, if we can.
Yeah.
Oh, almost
Yeah, that's
touching
it. Yeah, you got it.
Go
It's
ahead,
right there.
Tara.
Oh, I see it. It's one of the Stauropathes corals that was in front of a rock or something.
Okay.
Okay,
I
good.
see it now.
Yes,
Thanks,
on
Pilot.
top of the
Sorry,
rock.
false alarm.
Okay. Oh gosh, these things are gorgeous.
To push up.
Pushing up.
Okay, pilots, I think the ship is now in position.
Okay.
Copy that.
Look at that.
I know. Everything's changing so fast, I don't have time to scribble it down. Great layering.
Quite a crack there. from
Wow, we
top
have a,
right to
a
bottom left.
distinct crack or fracture across the screen from upper right to lower left, and it does in the upper right corner look as if the more coherent layer, the dark layer at the very top of the screen, kind of in the center, has been dropped down by what looks like to be maybe a meter along this fault, which is, if true, is a surprise, because that would mean this is what we call a thrust fault, where the left side has moved up relative to the right side. It is kind of hard to tell sometimes in these outcrops whether there's any coherency across fractures like these. But it sure looks like there's been some displacement on this particular surface.
How am I doing now, Don?
Looking good.
Okay,
I think
lots
we
of
could probably get
tails
another
coming
move
off
started,
these
Richard.
layers.
Maybe the same.
So because we're seeing a lot more tails
Yep.
covering
That whip coral
what were
before.
deeper
0,
down
20.
steps
Copy that.
between
Yeah, zeros,
these
2 0,
offsets
20,
in the
20
slope.
meters.
It looks as if
Bridge.
this is less well consolidated material. So
Jeff, I
that
know
just
we've
means
imaged that one.
that
Have you imaged
the
the one to the left?
horizontal faces that
Range,
we're
20,
seeing are more easily
bearing,
broken up
020,
and
the speed of
slide
0.2
down
knots,
the
please.
slope a little bit to cover up the step beneath.
Thank you, Bridge.
Say again, Roland.
I said, I know we've imaged that one straight ahead, but have we imaged the whip coral to the left?
I think we did. We did one just like that about 10 minutes ago.
Okay. I've been off camera for a few hours, so
That one right there you're talking about.
yes.
You can take a snap of it, but yeah, we have imaged. So hold that. It'll come up.
Wow. Wow.
Yeah, these are a lot more blocky than what we saw
Zoom some more.
below
So is
in
that,
terms of the rock.
Les was
Let's
thinking
look at
that's
this
a whip bamboo? And the what? Wait, what's the real fine one over to the right as well? Okay, look at that one first. Okay, can we
Okay,
look
we're
at the
looking
base
straight
of?
down on
Can
you,
we look at the
so
base of that whip,
It's got to be
please?
quick.
This one right here?
Yes.
Yeah. Okay, go ahead, zoom in some more, Rolly. I don't have much time though.
Drifting back pretty far. It's getting unlit.
Okay. Yeah, the current's pushing me back.
Yeah, you can definitely you can see the nodes. Ooh, I
Okay.
see stolen the first coral.
They're both scribbling like mad on the chat and in our notes.
Okay, we're looking back at
There's
you,
just
so you
so
need
much
to
to
move.
see here.
Okay, full wide,
Okay.
please.
Oh, did you want to see the base of that whip, or what was going? What was
Sorry,
going real
the
close to
train's
it? That's
moving.
okay. Yeah, that's all right, Jeff.
So it looks like a vertical fracture of some kind.
Oh boy, still in layered material, but
Jeez.
much more finely
Look at
fractured
this.
material, but
Jeez.
much more finely
Look at
fractured.
this.
The outcrop looks more blocky. Lots of right angle, actually 3 orthogonal angles to the fractures in a lot of the outcrop that we're seeing now.
There's a fish swimming on the
It
right.
is more or less
lasers.
horizontal, slightly sloping down to the
Very
right-hand
different
side.
with
And
those long
our
fins.
our
Oh, I
heading
got
is
it.
basically due north.
Okay, go ahead and partial.
Ooh, look, Shirley, a swimmer.
Okay, we've got it.
Fish.
Bruce calling our fish
Bruce
man.
Mundy,
like a
where
different
are
one.
Mundy.
you? We've
Have
got
you
the
seen
fish
that one?
Wow,
spotlight
can
up
I
in
see
the
that
air.
nice lateral line there?
Very distinct lateral line. White dots along the side of the fish.
How much time?
Okay, again, I have the lag, so I haven't seen it yet.
Maybe a
You
minute.
will. Wait for it. It's coming soon, and you'll see it even going
Yeah,
sideways.
You get a treat, Bruce.
Lasers are clear.
Okay, kill the lasers.
Yeah, Chris, if you
Sorry.
know what it is, shout it out.
Lasers off.
No way. I'm gonna let Bruce go with this. Go for this one.
Okay, from the shape of the lateral line, the way it sort of curves down in front and then has a short gap and then a continuation in a line along the side of the body, along with the shape of the gill cover, which has a sort of flap on top that's separated by a suture, and the shape of the stomach, which kind of bulges down and then has a protruding part at the end just before the anal fin. This is one of the live-bearing cusk-eels. You can see the filamentous pelvic fins placed up under the
tight on the face.
front of the abdomen, and the eel-like shape of the body with the dorsal and anal fins that are confluent with the caudal fin. So that's an ophidiiform. This is in the family Baphididae, the live-bearing cusk eels, and that gill cover morphology indicates the genus Diplocanthopoma. I'll type that into the event log in a moment. We saw some of these in the Hawaiian Islands. I can't remember if we've seen this genus in the dives here in the Marianas or not. I think we may have once. It's a very poorly known genus. Probably needs a lot of work with the taxonomy. But there really aren't enough specimens available to do a revision. There are two species in the Hawaiian Islands. There's one a darker brown like this, and then one a paler gray. This one looks a little different with the sort of bicolored grayish-blue head and the brownish body. So couldn't make a guess about the species. For all we know, it could be an undescribed species. Very interesting observation.
Is there another fish on the right?
Another fish?
No, I don't.
Lower center? White thing? Oh, no, maybe it's
No,
just
no. It's the top of a coral.
Okay.
Okay,
Just
thank you,
waving.
Bruce.
waving in the... Whoa. Right above the bottom of the screen, I'm seeing a series of radiating fractures that look like they're in a fan orientation. Completely confusing. Everything else we've seen looked like it had a series of orthogonal fractures.
You need to push ahead.
And this looks like just a
Or
sort of
up,
a
rather.
gentle change in angle radiating like a fan. And now we are back into the very well layered but less heavily fractured looking material that we saw further down the slope. There's some apparently very thin layers, likely of very fine material, between what look like stair-step blocks. And right in the middle of the screen, it looks as if there may be some offset in these for sure. It's not by very much, but it looks like there may be a fracture dipping to the west at a fairly high angle, down which the rocks on the left-hand side have slid by what looks like about a meter at most. Little bit of offset on this particular fault or fracture surface. It's got to be a fault if there's offset. Certainly looks like there's offset on this one.
Let's see, and look at that right underneath, like on the lower left. Wow.
Yeah, the lower left has a little bit of sediment, fine
Looks
sediment,
like we
just
might be
under
back
the
on that
overhang.
350
So I'm
again,
assuming
going
that
up slope.
whatever is making up these layers is breaking off and
Yeah,
collecting
certainly
beneath
direction
those
I'm going.
overhangs.
Yep.
Back to stair
I would
steps.
agree with the contours.
Okay.
We're at 1,672 meters.
Okay, Nav, you want to get another move underway? Another 20 meters? We'll try that 350.
Copy that.
And the top of the ridge is at about
20
1609.
meters.
So remember, Derek
3,
said that
5, 0.
it wasn't quite up to the top. Remember when he was showing
Bridge,
that
this is Nav.
Right.
other profile? So we may want to continue.
Hello,
Yeah, we
Bridge.
may.
Could I have
May
another
wanna
move,
continue.
please? This time we'll
Well,
have
what's that all in a row over there?
range of
Hanging
20
out
over on the
below.
left, over on the left-hand side at
Below
9
the
o'clock.
overhang.
2 knots, please.
Oh, this is so cool, Shirley. Hanging Gardens of Babylon.
Good
under
copy,
the
Bridge.
sea.
Thank you.
Okay, video.
Wow.
Oh my goodness.
Oh my gosh.
Are those anemones?
Our expert
pretty.
spotter, Annie, pointed those out. Oh my gosh. If we can get in a little bit closer to those, that would be awesome.
I have more zoom.
Let me try and push in.
Corallimorpharians, wow.
So what are they?
Oh,
So
I
I
knew.
see Lestis wrote Corallimorpharians, and I was going to guess that only because it seems that when we see Corallimorpharians in the deep sea, we often see them in these aggregations like this, which is kind of interesting. I'll just leave it at that.
Oh, but what are they? What is a Corallimorpharian?
A Corallimorpharian is something that belongs to the order Corallimorpharia, and that is in the subclass Hexacoralia, so it is an Idarian. And you can see it looks a lot like a sea anemone, which is in a different order of Hexacoralia, the Actiniaria. And there's been some debate. In fact, there was just a new paper published on the relationship of Corallimorpharians to the Scleractinians. Scleractinians are the stony corals, and Corallimorpharians have been described as naked corals, meaning they're essentially coral polyps that don't produce that stony skeleton. However, there's various genetic data that they have now that refutes that hypothesis. And one of the reasons that's important is because people have been looking at the effects of climate change, and one of the effects of climate change is increased pH of the ocean. And as that occurs, it may lead to, I should say decreased pH of the ocean, sorry, acidification of the ocean. As that occurs, it may lead to more difficulty in laying down the skeleton of a coral. And so there was some question about whether Arctinian corals could adapt almost like they had the genes of these Corallimorpharians to live kind of without a skeleton. So, you know, that's far-reaching and all conjecture, but the latest data suggests they're not all that closely related to the stony corals.
Wow. Thank you, Scott. You know, I really enjoy your comments on these animals because the classifications are so confusing to me, and I'll bet some of the people listening don't know either, but these guys sure look like,
They're beautiful.
yeah, anemones to me, but it's just fantastic to know more about them, so
Well,
thanks a lot.
At some point, we can have the discussion about relicanthus and anemones too, because I had been calling them anemones, but they're not.
Oh, yeah.
They're relicanthus.
Had no idea that there was so much diversity in these kinds of things.
All right.
Beautiful.
And
Beautiful, beautiful, beautiful.
of course, I would like to ask, why are they hiding under the rock? But, you know,
Full wide.
But it's
There's
another,
probably a
they can
reason.
hang on there.
To stay
Nice
out of the
space.
rain.
They're
maybe.
probably not getting sedimented on.
true.
They're still, you can tell by their
Sediment rain.
tentacles moving back and forth that there's still current there.
Well, actually, in the cracks, there might be kind of a concentration of current.
You think?
As the water
And
wafts
food.
across the outcrop. Oh boy, big stir sap.
Have we seen this coral? Assume partial. Hold back.
Oh my gosh, that poor
Thing on
that
the
poor black
right.
coral on the right.
It's
I wonder if
just-
something has gone and eaten off all of its polyps, and then this beautiful Chrysogorgia.
Okay, you can go. Zoom it, Will.
So this is what
Pretty.
we collected earlier, right?
Looks like it's the same. Yes, looks very similar to the one we collected earlier.
And has a shrimp.
Chrysogorgia earlier, and
Yep.
one of the things that we were remarking was that the branches
We're
had
curving
this
up.
smooth upward curve, and Patty, I think that is correct. They look the same. This one would certainly be an older colony, much more dense branches, and we're looking in the center there at a squat lobster, Chirostylidae is the family, and we often see these squat lobsters, these chirostylids associated with Chrysogorgia and some other octocorals, but very often with Chrysogorgia. And in fact, there's a lot of evidence to show that there
There's
are
150 meters to the top of
host-specific
the slope,
associations,
or
meaning
about
there are
210
lots
meters to the
I believe
dive end
there's hundreds
position.
of species of Chirostylid squat lobsters. And one of the reasons it's thought that is that because they associate with specific species of coral. And so it's very analogous to insects and flowers. Certain insects specialize on certain flowers, and so that may be one of the reasons to explain the diversity of those crabs.
Oh, thank you, Scott.
And the ship is in position.
Okay.
So, folks, we're about
We have about 2 hours left in our
yeah,
dive.
I'm just thinking we are about 60 meters below the waypoint 2, which was close to the top of this wall, but the slope of the wall diminishes significantly as we get to that depth, and what we were thinking about was trying to go a little bit further up. Well,
yeah, right. We were going to either go further up.
Yeah, either go further up or hang a left.
Right. So what? Yeah, what we were talking about.
Westward.
Right. In our pre-dive meeting this morning, we thought that we decided or discussed that once we got to this point,
we would make a decision.
We could make a decision about whether we wanted to continue to go upslope till we got to the top, or if we wanted to go along the slope.
Yeah, we're not quite to the depth at which we were going to make that decision. We've got about 60 meters to go, but start thinking maybe.
So, Nav, with that in mind, you want to get another move underway
Yeah, we're not
here?
seeing quite.
And let's go for 30 meters.
Can we slow down and get a good look at
That
it, please?
same
Yes.
heading, I
We'd
believe,
like to slow down and
350.
look at one of these sparsely branching corals. I think it's right in front on the right
30 meters,
lower corner.
350.
Is that
Roger.
correct?
I don't know
Is
which
that
one
true?
they're talking about. No, I don't see it. I don't. No, I don't think so.
I don't want to go back.
No, I don't think so. I don't think that's the one. I think that's just a dead.
Bridge.
Was it a?
Which one are you thinking about zooming in on?
Was it a bamboo or was it a black? It was bamboo, right?
Bridge could I have?
It was a bamboo, and we passed it.
another
Don't
Yeah.
go back.
Yeah.
It was a while
Okay.
back. That's
30
right. I'll let you know if I see
Bearing
another one.
350.
Okay.
It was kind
Speed
of
of
like,
0.2 knots.
I would describe it as a whip with two branches.
Okay.
Negative.
Range was 30 meters.
Let's just quickly zoom over to that thing on the left. It's at about 8 o'clock right now.
The kinky looking thing?
No, the one right above it.
Oh, in the distance?
No,
The-
close up.
The V shape.
The thing that kind of like takes a bend right up at the top.
Okay.
You think it has a little arm sticking out of it, don't you?
Yeah, I just want to. Well, no, I just wanted to make sure I don't pass. No, I don't think it's the one that Scott was talking about before. But
No,
I just
what
don't
I
wanna...
mean is it looks like it may have,
Okay,
it may
Tara,
still be living.
Let's go
Oh,
partial
yeah.
on it.
Whoops, it's lost its brittle star.
No, that's not it.
It's just
No,
a
that's
stalk.
not it. Okay, that's good. Thank you, pilot. We can continue.
There's a juvenile, what's it? I forget the names of these, Charlie.
Metallogorgia? Metallogorgia?
Yep, the one with the puffs.
With a brittle star?
So that's
Yes.
like 3 brittle stars that
Taking
bailed
a
off
break.
while
Is
we
that,
were watching.
I should look. Metallogorgia?
Metallogorgia, the one on the right-hand
Metal.
side?
Okay, metal. Why does it have the word metal?
I have no idea.
One of the things that the Commonwealth of the Mariana Islands wants to try to manage is its precious coral
Yeah, I'm trying to go down and give you a little more.
communities on the seafloor to figure out how many are there, where they are, what sort of substrates they attach to. So these dives are helping to fulfill that need for the Commonwealth of the Northern Mariana Islands within the monument area.
Tara, what are those pink things on the edge of this rock?
Looks like override arms to me.
Yeah. Let's just take a quick zoom.
Sure.
Yeah. He's on the other side. He, she.
He thinks he's hidden.
Okay, full white.
I can't see you, so you can't see me.
See another very red,
Oh, what's in the water?
maybe crinoid.
Missed it.
Center
So we
screen.
have a slope now. That
Oh, and then, but
is-
then look straight ahead. It's really,
Mhm. Oh,
wow.
yep, kind of steep up ahead of
Yeah,
us.
very steep.
Partial?
Is this a no? This is
Right, hold that, please.
all right. We seen one of these before? I think we have.
Was that the one that was the short stalk?
Nope, this has a long stalk. It's a very long
Yeah, I
stalk.
think this is what Chris was describing before, and you said it looked like a sea star at the base.
Yeah, I agree.
This has a longer stalk, though.
It does have a longer stalk. It is, I'm going to say it was
I
like,
think it's the same guy.
Yeah, this is the same one, and I can never see too many of these things. They're just so
I see.
beautiful, this incredible red color to them.
A Proisogornis?
Pro-isocrinus.
Oh, God.
It's pronounced Proisocrinus ruberimus. I didn't put the species name in. It's probably that species, but anyway, the ruberimus, I think, is referring to the red color.
Ruby. And there's our lasers for scale,
Great.
10 centimeters apart. So a lot of the layers
A
of
small little sponge there. I wonder if he was headed that way where he could image it. It's
on
to
the
the
right.
right of the lasers right now, a little vase sponge.
a little sponge to the right, just below that cobble in the center of the
my-
screen. You got the
is
lasers on it.
Euplectellid or something? I don't know, Chris.
I'm
Yeah.
gonna move
I
it
think
a
we
little
saw one a little
closer.
earlier, too. I think we both spotted it and made a note of it in the chat log.
Yeah, I've been distracted off and on today, so I very likely could have missed it.
No, I think you got it. I think you had it earlier, too. So the one we collected yesterday, Chris, that ring at the top, or that atrium, had a ring of spicules facing inward toward, radiating but inward instead of a sieve plate. It was very interesting. We didn't get any of the base of it.
Well, this could be the same thing.
Yeah, it's
As
looking
we're getting
like it.
closer, I would just like to confirm that because it looks like it has the overall same shape.
It does. It does.